So then we go to anaphase. Remember, at this point, the mitotic spindle are attached to kinetochores and they are shrinking toward opposite direction. They will keep shrinking toward opposite direction in such a way that the connection between the sister chromatids is going to break. As that connection breaks and the mitotic spindle keeps shrinking, they are going to part ways. The sister chromatids are going to part ways, right? So you see a group is moving down. Another group right here is moving up. And notice toward the centrioles, right? So here is a micrograph where we can see the chromosomes parting ways, right? Moving toward opposite direction. Here, this is anaphase. And finally, we have telophase. That telophase is going alongside with cytokinesis. During telophase, what we see is that the chromosomes will completely end up in opposite direction, right? The nuclear envelopes start reforming around the chromosomes. We have a cleavage furrow that develops and some protein fibers that are working toward pinching the two cells away from each other, right? So that they are going to see this line that develops. That line that develops shows you the pinching off of the cells away from each other. Now, understand that this distribution of the chromosomes has to happen concomitantly with the distribution of the cytoplasm, which is cytokinesis, right? Because before the membrane can close up here, you have to make sure that you have mitochondria here and here, you have ribosomes here and here, you have the Golgi apparatus here and here, you have what else? Lysosomes here, and all these organelles that are essential for the cell life, right? So that cell division that we just saw is that of animal cells. When we compare, though, the cell division in animal cell and in plant cells, we are going to see some similarities, but we are also going to see some differences. Remember that plant cells do not have a cell wall. Remember that animal cells do not have a cell wall, but plant cells do have a cell wall, right? Consequently, we have to take into account what is happening with the cell wall. So now, for whether you are talking about a plant cell or an animal cell, the interface is going to be the same, right? Same approach. The prophase is going to be similar. The metaphase is going to be similar. The anaphase is going to be similar. Where we are going to see a major difference, it's going to be during the telophase and cytokinesis, right? During the telophase, so here we are in metaphase, right? Here we are in prophase and prophase. So you see that the prophases are similar, no problem. Now, going into the anaphase, okay, we see anaphase here, right? We do see anaphase here, no problem, it's the same, similar. But then here we do have a cleavage furrow in animal cells, but in plant cells, we have what we call a cell plate. That cell plate, it's actually some vesicles that are carrying some fragments of the cell wall, because you have a new cell wall that develops along this area so that the two cells can part ways, right? So let's take a look at some details there. Now, maybe before we take a look at some details there, let's take a look at cytokinesis in animal cells. Cytokinesis in animal cell is going to consist of a cleavage furrow, right, that starts in late anaphase, right? Then we have a contractile ring. That contractile ring here is going to involve bunch of proteins, right? And these proteins are going to include the actin filaments, right? These actin filaments are the one that are going to trigger the pinching off, right? And then you have, you know, the moving away from each other, and you have a pinching off, and we have a graphic representation here. By the way, this is a micrograph of cells dividing, and this is another micrograph, okay? While here we have a graphic representation, and here another graphic representation. Now, when it comes to plant cells, we have this vesicle that are carrying some structures, right, that are going to actually define the plates, right? These plates actually are going to have the new membrane, the new cell wall, so that you are going to have two brand new cells that are going to develop. And here we do have actually the growth of the growth of that plate. All right. So again, the cell cycle is tightly regulated. I already told you about the G1 checkpoint, the G2 checkpoint, and the M checkpoint. What I did not tell you is what happens if the cell is not behaving properly well. I did tell you that the cell dies, but the death that happens here, we call that death apoptosis. When the cell, the integrity of the cell is not there, that cell is going to undergo apoptosis. Apoptosis is also called programmed cell death. Programmed cell death or apoptosis. So any damage that is extensive enough to cause an issue with the integrity of the cell is going to trigger the cell to undergo apoptosis. Now, let me also tell you upfront that what triggers apoptosis, it's the tumor suppressor genes. The tumor suppressor genes that technically say stop the cell cycle, right? They don't only say stop the cell cycle, they also trigger damaged cells to die, okay, by the process of apoptosis. Okay, so you already know the G1 checkpoint, the G2 checkpoint, the M checkpoint. We are not going to come back to that. Now, when a cell is damaged to the point that that cell is not worth to preserve, that cell will have to undergo apoptosis or programmed cell death. That programmed cell death is going to make sure that damaged cells are not staying where the body needs to stay healthy. That's one. Two, during the development, think for instance of the development of your digits, right? Your fingers. Now, when you look at the developing fetus, at some point during the development, there is a web between the fingers, right? Before you are born, all that web disappears. That web disappears because cells that exist between the fingers are dying. What causes these cells to die? It's that there is a signal that comes in that tells these cells, well, you don't belong here. You need to die. So they undergo apoptosis or programmed cell death, right? Another example, it's the tadpole tail. When you look at a tadpole, it has a tail, right? And over time, that tail is going to disappear. Where does it go? Well, that tail actually is lost via apoptosis, right? So apoptosis It's a process by which our system is regulating how many cells should be there or not. Okay? But now, there is another type of death that cells experience. That death is caused by external damages. For instance, when you have a cut, when you have a bruise, when you have something aggressive that comes from the environment, right? This is going to lead to necrosis. For instance, if you have sunburn, right? Your cells are going to die by necrosis, right? So typically what happens for apoptosis is that a normal cell is going to sense that there is something wrong. DNA is damaged. Well, if that DNA is damaged, then if we cannot repair that DNA, then we have to trigger programmed cell death. So typically here, the cell is going to put in place a mechanism by which the cell is going to eat itself from the inside. Okay? And after eating itself from the inside, that cell is going to be fragmented. Okay? And these fragments are going to be engulfed by white blood cells. Okay? That is a clean death. And you don't even know that something is happening. Right? You don't know that something is happening because it's part of the normal process of one's life. Now, we said that cancer is uncontrolled cell division. What can cause uncontrolled cell division? Remember, I told you about the proto-oncogenes being mutated so that they are overly active, so they become oncogenes. And then you have the tumor suppressor genes that become inactivated, right? So that they cannot stop cell division. I want you to remember that. But what can cause these genes to be modified? There are a number of factors that can be environmental, right? The carcinogens. The carcinogens are these conditions that can cause cancer development. That carcinogen are going to cause what we call carcinogenesis. Carcinogenesis is cancer development, right? That cancer development, it's multiple steps. It's not one single step, right? And that multiple step starts with an initial mutation in a cell. That initial mutation is going to cause the cell to unusually divide out of proportion, but not too fast yet. Nevertheless, it divides faster than an average cell of origin. So the cells are dividing and they are kind of behaving a little funny. Then the cells can experience another mutation. That's a second mutation. That second mutation is going to cause this mutated cell to behave even more weird, right? And unfortunately, over time, this already mutated cell can undergo another mutation. Now, the interesting thing in cancer development is that once a cell has undergone a mutation, the likelihood that the daughter cells can undergo a mutation increases. Interestingly, right? So you have a mass of cell. That mass of cell, it's actually what we call a tumor. Okay? Now, understand that this mass of cell, in order to thrive, needs food. Where is that food coming from? That food comes from the blood. Therefore, where you didn't have blood cells, blood vessels, you will have to grow blood vessels. That growth of blood vessels and lymphatic vessels is called angiogenesis. When you have development of angiogenesis in general, it's not a good news. It's not a good news. But let me explain that. The these cells right here, after undergoing mutation, 2 mutations, can experience a third mutation. That third mutation is going to seal the fate of these cells. Now, not only they are behaving weird, one thing I need to say is that cancer cells are not doing the job they were designed to do in the body, right? Now, these cells, not only they are not doing what they are supposed to do, they multiply out of control, they are also losing the ability to attach to each other. And as they lose the ability to attach to each other, and with the development of new blood vessels and lymphatic vessels, where these cells can actually enter the lymphatic vessels and the blood vessels to go in circulation. That's a really bad news. So we are going from a tumor, right? That is a mass of cell here, in as much as it is at that location and doesn't go anywhere, it's a benign tumor. When now the cells can get away from their site of origin, go in circulation, and go affect all the organs, we call this metastasis. That's a bad news. At that point, there is no repair possible, right? So here we're going to have cells that are going to unattach away from each other, right? They're going to go in circulation. So you can see how they are getting in the blood vessel and in the lymphatic vessels here, and go at other locations in the body and go cause damages there. So again, angiogenesis during cancer is never a good news. Then we have metastasis. Metastasis is a stage where there is nothing we can do anymore in terms of treatment. We can try, right, with available techniques. However, when cancer has spread throughout the body, it's just a matter of time before we actually die because of that cancer. Okay, so now I was telling you earlier about the proto-oncogenes and the tumor suppressor genes. The proto-oncogenes, their job, their normal job, is to stimulate cell division. But when they get mutated, then they stimulate cell division all the time, which causes uncontrolled cell division. Bad news. The tumor suppressor gene, their job is to stop cell division. When they are mutated so that they cannot stop cell division anymore, then that cell is going to multiply without stopping. Now, another thing that is worth mentioning about the tumor suppressor genes is that they trigger apoptosis when cells multiply out of control in a normal sense, right? In a normal sense, they can trigger apoptosis. But if they lose that ability, then it's too bad. It's going to be a cancer solution. It's going to be a cancer situation. All right, so here we do have some growth factors. Some growth factors typically are going to combine on the receptor, and that growth factor, upon binding on the receptor is going to trigger a cascade of events. That cascade of events is what is going to trigger cell division, right? And that's why you need actually the tumor suppressor genes and the proto-oncogenes to control all of that. Now, what in cigarette causes cancer? And cigarette is one of the carcinogens, right? And one of the carcinogens in cigarette bowl, you have a number of them. The most well known is nicotine. Nicotine. But nicotine is not the only one. You have a lot of other chemicals that are really a bad news that are in cigarette. You don't want cigarette, okay? Now, cancer can be associated with very specific genes. These specific genes have been identified. Some genes are, for instance, BRCA1 in the case of breast cancer, and BRCA2. Okay, these are some examples of genes associated with cancer, breast cancer. And we have the RB gene. RB is associated with the retinoblastoma cancer. Retinoblastoma is the cancer of the eyes, okay, because of a mutation in the RB gene. And then we have the P53. P53 is another gene that actually, when altered, can cause uncontrolled cell division. And also we have the RET gene that also can cause cancer, that is associated with cancer. Now, there is another case of cancer that I would like to address that I've not addressed so far here. That cancer is caused by translocation of chromosome pieces. So we have chromosome number 9 and chromosome number 22 in humans, right? This chromosome number 9 actually carries a gene called ABL. Chromosome number 22 carries a gene called BCR. Now, we see in the misdistribution of chromosomes that there is some translocation of the chromosome that can happen. In other words, there is an exchange of genetic information between non-sister, non-homologous chromosomes. Non-homologous chromosomes. This non-homologous chromosomes, it's 9 is 9, obviously. 22 is 22, right? And then they exchange their legs. When they exchange their legs, so you see that a fragment of 22 have come to 9, and a fragment of 9 have come to 22, so that BCR gene and ABL find themselves on the same spot, so that the expression of this gene is going to be interdependent. And this is the case of what we call the Philadelphia chromosome, when chromosome 22 is modified with a translocation, and that BCR-ABL translocation is responsible for 95% of the chronic myelogenous leukemia. It's a form of blood cancer, right? So now, finally, but not least, let's take a look at breast cancer. So here we do have a micrograph of cells in which you do have cancer cells that are right here represented in orange, yellow. These are cancer cells, while in green and blue, the blue being the nucleus, right, you have normal cells of breast cancer. So breast cancer is going to start as a benign tumor. That benign tumor is going to grow, right? It grows so that you are going to have blood vessels that are going to develop, and lymphatic vessels that are going to develop, right? So here, the cancer has grown so big that it's now called a malignant tumor, okay? That malignant tumor is now going to have cells that are going to go in circulation, and when they go in circulation, they go throughout the body. That is a bad news, and that is a point of breast cancer that you cannot treat anymore. You can attempt, but it's difficult, right? So that's basically the development of breast cancer. But what can we do to prevent cancer? We talked about, okay, well, cancer, how it happens, but what can we do to prevent cancer? To prevent cancer, well, we have to basically live a healthy life, right? That healthy life requires making some lifestyle choices, right? Maybe before we dive into those lifestyle choices, let's first of all address the treatment, because when you have it, you have to treat it. The ideal situation for a cancer, any cancer, is surgical removal of the tumor. If this is not possible, this is when you have to use other techniques, including radiation therapy, chemotherapy, and immunotherapy. Now, in all the cases, radiotherapy, chemotherapy, immunotherapy, this is relatively heavy on the patient. Okay? Of all the treatments, the treatment that is the least heavy is the surgery, right? Even when it is taking place in the brain, it's better than having to undergo radiation therapy, chemotherapy, or immunotherapy, because anytime you go through these different therapies, chance that normal cells are going to be affected is very high. Why? Because our cancer cells share a lot with our normal cells than they exhibit differences. That is a very important aspect to know. That's why it's so difficult to treat cancer, right? A cancer cell is not a bacterium. A cancer cell is not a virus. A cancer cell is not a parasite, right? Which are structures, biological structures that are completely different from our cells, right? So now going to the prevention, okay, avoiding a number of things, including avoiding the use of tobacco. No matter how you turn this, tobacco is not a good news for you. Limiting the use of the consumption of alcohol, avoiding sun baths, right? Exposure to carcinogen can be a problem, okay? Such as the radon gas. In some places where you have a basement, you have to be careful because those basements can accumulate a gas that is called radon. That's why in modern housing today, you have a little device that can detect the amount of radon, right? Do exercise. Exercising is very good to preventing these conditions, and also have a really nice diet with a lot of antioxidants. Eat a lot of fruits that are going to contain a lot of antioxidants, right? That actually prevent you from getting cancer. Finally, review the chapter at the end of the chapter, at the end of the book chapter in your book, right? There is a review there. You can also write your own review, okay? Assess your understanding of the material. Do some questions that are at the end of the chapter in your book, or design your own questions. Talk with your classmates. Ask them if they have some questions they can share with you, and also volunteer to share your own questions, right? Because, you know, to support each other, you have to participate, right? And finally, create a handwritten concept map, okay? That can be very helpful. This is what I had to share with you. Take care.